Unlike what is written on your agenda or schedule, I will try to summarize the research studies over 55 years of discovery for you and present some current perspective. And so that's the first part of my talk, and the second part will be shorter, but about some information about funding. You heard a lot about clinical this morning, but just put on your research hat, and because I'm a researcher, I don't see patients except when they come to the research unit or when they used to come to the research unit at OHSU. But researchers, they have a different view of the world than clinicians. And they not always answer the questions that you would like to have asked about, is there a cure? What can I do to solve this? Researchers, they ask questions and they, I would say, rarely find answers. They just open up more questions. So I put this, on this picture that I got from the web. And what kind of number do you see on this picture? Would anyone tell me? 71, 74. Anything else? Well, what I see is 21. So can you see 21? No, so that's just to illustrate what the research process is about. You have several investigators, several researchers. They look at the same thing. They look at SLOS, for example, and they see different things. They have different interests and they have different perspective. Here, this is, I can see 21 because I have this genetic disorder which is called color blindness. Nothing bad, but I don't distinguish very well between red and green. So, but that's the research process illustrated. I think I need to use that, right? I tried to illustrate the research process using Pubmed. Pubmed is a public database that where you can find all what has been published about a given topic. In that case, it's SLOS, Smith-Lemli-Opitz syndrome. I plugged that in the searchable field, and I came up with and you can do the same thing yourself. It's accessible to everyone. With the number of publications, which I illustrated as a cumulative account of research publication. And you can see, hopefully you can follow that. It started in 1964, which is about 55 years ago. So kids diagnosed with Smith-Lemli-Opitz syndrome 55 years ago would be 55 years old now. So what happened since then, you had a slow increase in the number of publications, and until 1993. 1993 is when the metabolic defect was discovered, as you heard earlier. And at that time, the number of publications rose dramatically. And you see it rose continuously until present, which is 2019. The scale goes to 220, but it's 219. And there are about 850 publications. What you could see also, if you look really with a researcher eye, it rises very steeply there. And around about 2010, there's a slow inflection of the number of publications. And that slowing down can be better seen with this graph here. So this is a density graph, basically by year, and it's by every 2 years. And you can see, again, the slow rise, and then a steep rise. And starting in 2000, 2005, there's kind of a slow decline in the number of publications per year. So you could say, well, people are not interested in research at that time. I don't know the cause of that, but it could be multiple causes. It could be because funding declined, because studies become more in-depth and take longer to accomplish, fewer researchers involved in the process of studying SLOS, but there is a decline in the number of publications. By comparison, I wanted to bring up other rare diseases. This one is called SSADHD. I will pass on the term, but it was discovered in 1983, and there is also a beginning of a slow rise, as you see in the number of publications, and a start of a steeper rise. Right now, it's at 130 publication total. There is another one here that I want to bring up, which is Sjogren-Larsen syndrome. Dr. Rizzo might tell you a lot more about it, but it's followed pretty much the same kinetics or rise as SLOS, although it reaches now with a total of 400 publications since 1983. And now we have the extreme. This is the group of conditions, rare disease conditions called lysosomal storage diseases, and it was discovered and described in the early or mid 40s, and you can tell that right now we are at 27 or 27,500 publications. LSDs are a group of, I think, 60, let's see. Yeah, 60 different disorders. So you could say, well, there's a lot of activity and research activity because there are several conditions. But among those, you have one condition, Gaucher disease, that also was reported in the mid-40s, and that now totals 5,500 publications in 2019. All this is from Pubmed, all right? I haven't read those publications, or except a few of those. So what? So the number of publication and research effort may also depend on the prevalence of the condition. The SSADHD, the prevalence is 1 in about 2 million, so it's ultra rare. SLS, I think maybe Dr. Rizzo can tell you more about it, but I estimate it at 1 in 250,000. That's maybe 300,000. For LSDs, the Gaucher disease is 1 in 60,000 about, which is the top range of SLOS prevalence, or the lowest range in prevalence of SLOS. But there are some LSD conditions that are extremely rare, like this sialidosis. Will make it 1 in 4 million about. So the reason I'm bringing this up is to show you the trend, but also the example of lysosomal storage disease, to me, is telling that if you pool several conditions, rare disease condition, and have a critical mass, that may drag and attract more research and research funding and research interest than if you are just by yourself. And so my simple message here is maybe as the SLOS families might consider working with other groups of rare diseases, not necessarily related to sterile disorders, although that's what we try to do with the STARE consortium. But that may bring up more mass critiques and attract more funding if you can pool your resources together. So now we'll get a little bit deeper in the analysis of those curves that I showed you, but staying and focus on SLOS publication. So that's a nice, colorful graph. I cannot even tell you which colors there are because, as I told you, I'm colorblind. But I try to, don't quote me, but I'm lying here. I tried to read all those 850 publications. in depth in a couple hours maybe, and try to categorize them into broad categories of interest. So one is the clinical interest, publications focusing on genetics, others focusing on metabolic and signaling, others focusing more on treatment, and others focusing more on oxysterol and oxidative stress. And this graph shows you, again, this trend in, it represents the 3D, that's by decade here, and that's the number of publications. You could see the trend again that peaks in the around 2000, 2010, and then declines afterward for pretty much all categories, except one here, that's the one I hope, which is the category of oxysterol and oxidative stress publications. I brought those two together because there is a link, metabolic link between the two. This seems to be the only category that seems to rise steadily in terms of publication, which reflects research effort. The other notable point in this slide is that there is very few, if almost nothing, in terms of gene therapy, which is this little, the far right row or column here. So I'm going to go now a little bit more into, illustrate some key steps in SLOS research. And the point here is not to tell you again what you heard yesterday and some of it today, but is to give you an idea about the timing and the time it takes to go from one concept or one discovery to another. From a research perspective, I should say that it goes very fast and progress has been steady and impressive. Maybe from a patient standpoint, it's very slow. But that's what it takes to go from one point to another point. And so that's the research perspective that is a slightly different compared to the patient perspective or the family perspective. So you obviously all know that in 64, that's the first clinical report. 93, cholesterol deficiency was identified. 1998, that's the cloning of the gene. And the description of SLOS mutation in that gene by Chris Wasif, whom you heard yesterday. There is also in 1998 the association between 7-dehydrocholesterol and photosensitivity. And I will get back to that a little bit later. 2001 and 2006, you have the reports of the animal models that Dr. Porter, his team, and Chris described and told you about. And all this contributed to identify the metabolic impairment that underscores the condition. I will not go into the detail, but it's really about the imbalance between not enough cholesterol and too much 7-dehydrocholesterol. So we stopped in 2010 on the previous slide. Now, from 2010 to present, there have been a lot of reports and activity that has gone on. And I have to beg forgiveness to those in the audience who have contributed to that research, because I put only just a few sliver of what has been done. But to me, those are important points that may have relevance to treatment, cures, and your expectation about research. So in 2010, I think there is a dramatic, significant turn about on the research about 7-dehydrocholesterol, and that's the identification of the oxysterol and the beginning of oxysterol research. The oxysterol are formed derived from 7-dehydrocholesterol, and they are toxic to cells. And that paper, those images are from Dr. Corradi, who's been invited at the previous, the Cincinnati family meeting, and that shows the toxicity of those oxysterol. And I won't go into the detail of the experiments, but here you have cells that are kind of neuronal cells, not treated, but treated with oxysterols. And you can see the toxicity just with your bare eyes, naked eyes, I guess. The number of cells is dramatically reduced. In 2016, that same group and others and collaborators reported a new way of screening for small molecules that increase or modulate the concentration of 7-DHC. And that screening is a cell-based screening assay. And so that's the concept of modulators, 7-dehydrocholesterol modulators. I will get back to that a little bit in more detail in another slide. But that's already 6 years after 2010. And ongoing, you have a number of tools that have been developed for screening of small molecules using fibroblasts, skin fibroblasts from your children, zebrafish, as Chris Wasif mentioned earlier in this conference, modifier genes with the CYP27 that Dr. Porter and Chris mentioned yesterday. So that sequence here is quite dramatic, and there's a lot of promising data and concept that came out of this. So now, also during that same period, there was some interesting discoveries, and not all from investigators who are focusing on SLOS, but in 2015, there was a group described the crystal structure of an enzyme, a dehydrocholesterol reductase, that is closely related to DHCR7, and it's DHCR14. And that crystal structure is represented here. So you see, this is a lot of rods and connected by little wires, basically, and that's DHCR7. That's an in silico model of the enzyme. That report and that work led to the identification of amino acids responsible for cholesterol-mediated regulation of DHCR7. That's a paper that was published in 2016. And then a year ago, there was a report of in silico modeling of DHCR7 and the impact of describing the impact of mutations on that molecule. So this is what is represented here. You have good eyes, you can see red and greens. I cannot see anything on this. The bottom line is those little loops and connective stretch of amino acids here. That's a way of representing how the molecule, the DHCR7, function in the membrane. And when you can put this in a computer, model that molecule, and you add mutation, you change one amino acid there, another one there, you can estimate how stable the molecule is and how unstable it becomes because of the mutations. So to me, that's a very important step in SLOS research, because then you can, what researchers call, you can dock small molecules to this computer model of the enzyme and find molecules that can restore the enzyme and the stability of the enzyme and maybe improve its activity. So that's a really, to me, that's an important step in SLOS research. Now, we're still in that present decade. I think you heard from Kevin this morning about the iPSCs and the stem cells. Well, he mentioned also that in 2010, our group, in collaboration with the Dr. Porter's group, We isolated neural stem cells from mouse, SLOS mice and wild-type mice, and we differentiated those neural stem cells, isolated from the brain of those mice, into neurons. And I think you can easily see the difference in aspect, in phenotype, as we call it, between those two strains of mice. Importantly, the mutant neurons have a lot more extensions, are bigger, and there are fewer. You don't see that. But this is a nice tool also that we had planned to use to screen for molecules and other things, small molecules. But we used it also to identify some signaling mechanism. And here is the difference in response of those cells to glutamate. And glutamate is an excitatory molecule. When you have too much glutamate in your brain, it stimulates neurons. It increases intracellular calcium, which is what those graphs show. And it has, it increases the excitability of the brain. So that's one of the tools. And I don't want to summarize in just one panel here on the right side the years of research that Kevin presented this morning. But this is that's a picture that illustrates the difference between fibroblast-derived iPSCs from control and from SLOS patients. And again, this is red, I suspect, but you don't see any red here, so that's the difference. Is that red, Kevin? Good, good. I'm good on that. So I won't dwell on that, but that came in 2016, and there is ongoing research, as you heard this morning, about this very exciting research. Now, I step back a little bit and wanted to show you a few sequences of events that led to the cholesterol and bile acid supplementation studies. I won't go into the detail too much, but it's just, again, to give you a perspective of the timing. So in 1994, which is really right after, or at the time the metabolic defect was identified, there was an attempt to design oral cholesterol supplementation for patients. The treatment with cholesterol and bile acid was proposed already in 1996, 97. There are a few studies. Some of them showed no improvement with cholesterol. So again, I want to stop here and say I'm not going to tell you if cholesterol is good or bad. I'm just telling you what the sequence of event and where we are with the research. 2007, there was a report of partial rescue of retinal abnormalities with cholesterol in the rat treated with an inhibitor of DHCR7 by Dr. Flissler. I think that helped and conditioned Dr. Elias to work on the retinal aspect of SLOS. Here, I'm not mentioning natural history studies, but I wanted to make a point about the 2009-2014 cholesterol supplementation intervention study that the STARE consortium had initiated. And I mentioned about it by NICHD, which is actually true. NICHD asked us to stop the study in part because they thought that we had not recruited enough patients in our study. So maybe there were some other considerations I will not talk about here. But my point is that we had enrolled about 21 patients in that study, and a few of them came back for a second visit. It was a longitudinal intervention study, and we had trouble bringing in families over and over regularly for longitudinal evaluation. So as families, and if you think about doing research or getting engaged in research, especially the natural history study, where you come at regular time, it's very important from a research standpoint to stick to the schedule as much as you can, obviously. So in 2019, Dr. Elias started, or will start soon, yesterday, tomorrow maybe, this bile acid treatment or intervention study. It's a pilot study that we hope to complete within a year or so. Now, just to finish on that topic here, I mentioned I want to put in the blood-brain barrier permeable cholesterol analog studies, which is really in its infancy, that Dr. Steiner presented yesterday. In terms of cholesterol supplementation, you have obviously the egg yolk, the suspension, and those analogs that Dr. Steiner talked about. One thing that has not been talked about is the nanoparticles, and I don't know the details of the Neurodel project exactly, but there's a great deal of research on encapsulating lipid molecules, small molecules, into tiny nanoparticles the size of LDL cholesterol, basically. And those nanoparticles can be directed to specific tissue in the body. And it would be nice to have studies developed in that for SLOS with nanoparticles that could target the blood-brain barrier, penetrate, and supplement and replete the brain with cholesterol. This is the cholic acid study. As you know, when you don't make enough cholesterol, you don't make enough cholic acid, and the cholic acid deficiency, in principle, decrease cholesterol absorption by the gut, and that contributes to further increase the cholesterol deficiency. And the bile acid, or we hope, will be fixing or rescuing that cycle here. Now, there are other cholesterol supplementation studies that have been considered. In 1999 already, so we're talking about 20 years ago, that if I count right, there was antenatal therapy with fetal plasma transfusion, IV and intraperitoneal of fresh frozen plasma. So that was in 1999. 2014, there was a transfusion of fresh frozen plasma in SLOS newborn this time. And in 2019, I saw a report, and I cannot really gauge the value of that report, but it's a liver transplant in a young boy. So the liver, transplanting a functional liver or a piece of liver with the functioning enzyme DHTR7, the authors claimed that there was a remarkable increase in metabolic profile. That means increased cholesterol, plasma cholesterol, 7-DHT going down, and an improvement in fine motor skills. What is interesting in this study, and that's the reason I brought it up, is that as for many transplants, the drug that is immunosuppressive is the tacrolimus or tacrolimus. Tacrolimus is an immunosuppressive drug that raises cholesterol. So that's quite interesting that it may have played a role in the improvement, besides, in addition to the liver part. Some studies also suggest that the drug improves autophagy, and autophagy, at least that's been demonstrated in cells, autophagy is a mechanism, a cellular mechanism that improves the clearance of debris in the cell, and just put it simply. And as you heard already, you have oxysterols that create damages, you have oxidative stress, and maybe that drug has some magic power to remove those debris and clear and improve the clinical symptoms. This is pure speculations from my part, but this is where the research mind goes, at least for me. Now, antioxidant studies, again, we have a long series of years that started in 1998, and I think already in 1998, the skin photosensitivity that some of you have experienced and continue to experience was associated with the elevation of 7-DHC. And 7-DHC is unstable, and that is what is the 7-DHC is the precursor of oxysterols in some ways. So a series of steps here, discoveries of oxysterol, oxidative stress markers, and that led to antioxidant supplementation in cells and in mouse. That's a study in 2013 by Dr. Corradi in Ned Porter's lab, I think, at that time. And she identified in this study vitamin E as being a key component of the oxidative stress antioxidant property of the antioxidant supplementation. And as you know, we have a study with cholesterol and antioxidant that is going on, that has been going on for quite some time now, that was presented by Dr. Elias. Dr. Corradi told me that there was an ongoing study in Hungary, so I don't suggest you go there, but you know, and it's a 3-year prospective study with only 6 patients, so that's not many, but it's with vitamin E as antioxidant. And apparently, she says that she communicated to me that there were some positive clinical signs observed in half of the patient. Half of the patient is 3 out of 6. So here we're far from having a powered study, but you know, everything is encouraging. All right. Modulators, 7-DHC modulator study, I will go quickly about this, is the Simvestatine concept that originated already almost 20 years ago. That was proposed by Dr. Gira, you say that, or Gira, in some ways. And you heard about the results of the Simvestatine study earlier today. So this is just the mechanism of action. I will pass on it because it's been explained in details. So now, still about 7-DHC modulator studies. This is the loop that we're considering here, 7-DHC, accumulation, oxysterol, and damages to the cells, cellular toxicity, oxidative stress. So there's a series of studies that started in pretty much in 2005, 15 years ago or so. This report showed that some drugs were capable of modulating the activity of DHCR7, the enzyme. And those drugs are antidepressant, antipsychotic drugs. And you have a few names here. That was back in 2005. And then another study in 2010 that came out. 2013, there were a couple of reports expanding that line of research with antidepressant. You have Abilify that you heard this morning, and Trazodone as well. Trazodone is one of the drugs that we heard that could be used for sleep studies. Abilify also is one of the drugs that the long-term drug that could be used. And those drugs have been identified as increasing 7-dehydrocholesterol. So I'll make a point about this in a second. But in 2016, there's another paper by the Corradi Ned Porter group that reports the screening of small molecules capable of decreasing 7-DHC cholesterol in DHCR7 deficient cells. So this paper is important because it's one more tool that was developed to screen molecules that may reduce 7-DHC and reduce oxidative stress in SLOS. And there's this other paper with new drugs, antipsychotic, antidepressant drugs, that increase 7-DHC in cells and mice. So the important point here also is that not only you have a system to identify molecules that can decrease 7-DHC, but you identify molecules that may have a toxic effect or side effects because they increase 7-DHC. And those are the studies that I selected to show. One in 2017 that showed that carriers of the SLOS mutations might be more susceptible to those psychotropic drugs that increase 7-DHC. This is research here, and that's in vitro research. Doesn't mean that as a parent and as a carrier, you cannot take those drugs, but that means that there might be research needed to further explore what it can do in carriers. And there's this 2019 report, again by Corradi and colleagues, that show that maternal exposure to psychotropic drugs that increase 7-DHC can alter embryonic development. So those are drugs that, these are mouse studies, they are not human studies, but that again raised the possibility or the question of, you know, how should those drugs be administered to carriers, obviously. Take-home message for those modulator studies, that's the big thing, that the number of publications that has been increasing steadily in the past 10 years about. Simvestatine may not provide significant clinical benefits, and I'll let Dr. Porter's last word earlier to stay and give you more information, obviously, on this. Some drugs used in patients or carriers increase 7-DHC. So the clinical toxicity, that's in research studies, but the clinical toxicity of those drugs need to be carefully examined. But other drugs may decrease cellular 7-DHC, and that's very promising. But again, the potential clinical benefits need to be evaluated. So other research with therapeutic potential, we have gene therapy. I told you about four papers only, and all in the mouse. So nothing attempted in humans at this point. There are a couple other therapeutic targets that have been proposed or that are under exploration. I think the one to remember is the Wnt beta-catenin pathway that Dr. Francis is investigating at this point. That could be a therapeutic target in some ways, but obviously, as he pointed out, it will be several years before something comes out. Or maybe tomorrow, actually. We don't know. He needs to go back to the lab tonight. The ABCA1 transporter is something I will pass on. There's something that I haven't seen in the literature, and that may be under investigation, and I don't know about it, but it's the gene editing concept. So gene editing, as you heard also from Kevin earlier today, is about just changing the mutation using some techniques. He's used that in vitro in cells, but people have done that in vivo in humans as well, in the mouse, obviously, but in humans. And this is two studies that are going on that were communicated to us at one of the last Rare Disease Clinical Research Network steering committee meeting maybe half a year ago, I think. And those are gene editing studies that are undergoing for mucopolysaccharidosis conditions, the 1 and the 2, and those are also rare diseases. And basically, it's injecting some viruses that contain enzymes that will cut and repair the DNA in vivo. And so I don't think there's anything published in terms of efficacy, but it's probably mostly a feasibility concept that is tested here. We should hear more about this. The important point here is that SLOS is one thing, but the research on SLOS gained from any advance that is made in other fields and other rare disease condition. And it's, again, that's a message to consider maybe partnering with those other rare diseases and patient advocacy group to gain from them and learn from them, not only in terms of funding, because that's always number one, but in terms of ideas about attracting scientists and so forth. So now, just 2 or 3 slides that are very dense, but this is what you can find on this website, which is a public website, clinicaltrials.gov, and you can see the studies. If you type in Smith-Lemli-Opitz syndrome, you can see the studies that are ongoing or the studies that have been completed or terminated. You can see on the top Dr. Elias' study on cholic acid that will start very soon. You have the cholesterol and antioxidant study. Those are registered studies, obviously, again by Dr. Elias. And you have Dr. Porter's longitudinal natural history study listed below. You have a few completed or terminated studies below. Some of them you will see were about dietary cholesterol and the effect on patients. The Simvestatine study that Dr. Porter and Chris Wasif reported earlier today, and some others. You also have my favorite one that was terminated in 2015, which is the OHSU STAIR dietary intervention study. So you have some contact info, but the best is to go to that site and check what is available. Now, here you have also information from public database, which is the Reporter Database, NIH Reporter Database, that lists the studies and the institutes that funds them, the PI, the principal investigator, and the amount of dollars that is given for that particular year. And I think those are numbers for the 2018 year, maybe, or the current fiscal year. Don't quote me on this. But that gives you an idea about how much money is spent, at least by NIH, extramural or intramural here on SLOS research. It may look like a lot, but actually it's not a lot. I can't go in the details of that, at least for some studies. But you notice that there is studies on neurogenesis and environmental agent by Ned Porter at Vanderbilt, neuronal and environmental stress also. You have studies on oxysterols going on, some studies on should I say morphogenesis or embryonic development and malformation. The STAIR consortium also is getting money, but it's not for SLOS. It's mostly for other rare diseases. And those are total costs here. So think about $600,000 for 1 year. It's about $400,000 direct cost to support research and a consortium that has 5 or 6 sites. So it's quite small. You have other projects on gene function, expression, and regulation in zebrafish. I think Chris Wasif referred to that as that's not the whole pot of money is not just for SLOS, but that has been instrumental in developing a zebrafish model for screening small molecules. So that's where the taxpayer money is invested, which is really good. clinical investigations. I believe this must be Dr. Porter's natural history study, right, Denny? I think that may be it. And you have a couple other, and you have Kevin's grant and support to continue the study on induced pluripotent stem cells. Now, just a brief overview of what the support of the foundation of the families has been over the past decade. I cannot get before that, but I'm sure the foundation has supported research before that. But currently, there are three projects that are supported. One on steroid phosphoesters, called the main molecule, parent molecule is ionotropin, which is an analog of spironolactone. And spironolactone is a steroid molecule, basically, that regulates water and potassium homeostasis. So there is one project on this. There is support also for Kevin's lab to further investigate iPSCs and the screening of small molecules, the effect on the Wnt beta-catenin signaling. And there is support also for Dr. Steiner and Neurodel project on developing small cholesterol or analogs of cholesterol that may penetrate the blood-brain barrier. Here I just summarize the grant review process. So it's about significance, feasibility, and likelihood of long-term funding. So that's what the foundation is doing, encouraging research that can lead to further investigations and long-term funding. And you have here a list of other studies that have been funded in the past, that are completed over the past 10 years. Now, I just want to finish with that slide. I hope I'm not too over my time. But this is a slide that has nothing to do with SLOS, but it's a summary of a study that the STARE Consortium did with another group of rare disease called peroxisomal biogenesis disorders, specifically mostly in Zellweger syndrome. And the study was initiated, conducted, and published as first author by Dr. Bose Musumi, who is also the parent of a child with this devastating condition. So here you have a parent who is engaged in the research process, who's been challenged with that condition very personally, and who put her effort, I guess her husband is probably in that process as well, but dedicated her life to research on this condition. So she did this study on the emotional experience in parents of children with Zellweger spectrum disorders. And it's a qualitative study. It's a focus group study where parents were invited to share their experience, emotional experience with the condition, the disease. And it's about parents this time. It's not about children, it's about parents. And here you have some quotes from the abstract. This is a paper that you can get from the web. It's public. So nearly a third of ZSD, that's the condition caregivers, describe their overall emotional experience as a roller coaster. So who has not experienced roller coaster emotions with SLOS? Feeling overwhelmed and devastated were the most frequently described emotional responses. The most common coping strategies were acceptance of limitations of the diseases, redefining normal in the parenting experience, and advocating on behalf of the child and the patient community. So those are really quotes. It's quotes from parents that are summarized in those two small paragraphs. And the study underscores the profound emotional impact on parents who are caregivers for children with ZSDs. And that highlights the utility of patient community feedback and qualitative approaches to fully characterize the overall family experience. So this is to me, this is an approach, a research approach, that could interest families with SLOS. I think it's important to capture what parents experience. That's from a personal standpoint, obviously, it's good, but it also raises awareness of the condition and what it takes, the toll it takes to caregivers in particular, so that you can get better care in return. So having a research study that really shows what you experience will go a long way in improving the care of your children. I think that's my take on the study, and maybe Dr. Rizzo has an additional take or a different take. So this is my last slide here. Some take-home messages. So those are my take-home messages, and you can take whatever you want. There has been tremendous progress over the past 55 years on many fronts. Clinical, molecular, metabolic mechanisms, the understanding on how the condition what underlies the condition at the cellular and molecular level. Been identification of oxysterols as potential therapeutic targets. I think that's the next new thing that may come out with some maybe direct and significant impact on families and patients. And also the development, more recent, of cellular and animal-based drug screening tools. That's pretty critical now that people can do high throughput screening of small molecules, investigating 700,000 molecules at the same time. I know we're not there yet, but that's the goal, and there may be some hits there that would be useful for patients. Still, so that means this research effort needs continuing support, and it needs continuing support from families and obviously from researchers. And so there is a need to support studies on pathogenesis. There is still a lot unknown, and I think Dr. Francis illustrated this very well this morning. And there is a need to support investigations for finding new drugs, not only that work in the test tubes, but also that work clinically. Not only in decreasing 7-DHC, but improving clinical symptoms. So there is a need to continue that support, but we are still fraught with, I think that's the word, with no comprehensive characterization of SLOS natural history study. That's why the study that Dr. Porter has continued and sustained for years needs to be continued with your help. So there's a need to further engage the patient community in natural history studies. Everybody should participate. That's my view from the research standpoint, obviously. I don't have to deal with an SLOS child. There is no cure, no targeted therapeutics, and there is still uncertainty about effectiveness of cholesterol supplementation. I think you heard over and over again that it's everyone receives cholesterol supplementation, but there has not been a controlled trial that proves definitely that it works. Hence the need to, again, support studies that will show that. And the trend toward decreasing SLOS research publication in the past 5 years could be worrying, but I don't want to leave you with the idea that you are abandoned from a research standpoint based on this number of publications. I think the research is really thriving, and there may be ups and downs at some point depending on, again, funding, investment, younger investigators starting their career and coming back to the SLOS research. But the message here that I would like to emphasize again is, as families, I would encourage you to team up with other patient advocacy groups representing other rare diseases, either caused by steroid disorders or other rare disorders, to increase representation and raise more funding. Really, we discussed this yesterday at the board meeting. Funding is le nerf de la guerre, as we say in French, which is what supports everything. So without dollars, research cannot happen, and there won't be any cure. So with this, I think I took too much of your time, and if you have questions.